Olá, boa tarde a todos que nos assistem. Hoje nós temos a grata satisfação de contar com a contribuição da doutora Cheryl Andam, que é professora assistente da University at Albany, a qual constitui o principal campus da State University of New York. Para aqueles que não sabem ou esqueceram, como eu faço frequentemente, Albany é a cidade capital do estado de Nova York, não a cidade de Nova York, que é bastante badalada, né? Então, ela tem todo um contexto histórico e ela tem também um foco universitário muito importante, além de ser uma localização muito interessante por vários motivos. A doutora Andam, ela lidera um grupo envolvido no estudo genômico de bactérias patogênicas, sobretudo aquelas ligadas à resistência a antimicrobianos, e ela falará um pouco sobre alguns aspectos da sua pesquisa e sobre as oportunidades que ela está oferecendo para colaborar ou fazer parte do grupo dela, e também as oportunidades que a University Albany oferece. Né? Well, after this short introduction in Portuguese, I would like to tell Sherry how pleased we are in having her with us today. And she'll be talking about her major interests in research and also be discussing about the opportunities that she has to offer for people interested in joining her group and also collaborate somehow with her. Cheryl, again, many thanks for selecting us as a target for your presentation. And go ahead.
Yes. Thank you. Thanks, Lucia, and also very grateful to Stephanie for organizing this. So I'm going to talk. So there would be two components of my talk: a short, introduction about our graduate program, and then I'm going to talk about a recently finished project that we did, and hopefully this would pique your interest in our research. And just want to let you know that so I've been a faculty member in two institutions for the last 6 years, and I want to emphasize that I've been a member of the departmental graduate admissions committee in my previous institution, University of New Hampshire, as well as in SUNY Albany. So I'm a member of the Departmental Graduate Admissions Committee as well as Recruitment Committee. So that's why I'm really excited to give this talk, because I really want to encourage you, your students, to consider our graduate program. And so my research, just to give you an overview of what we do, we work on genomics, genomic epidemiology of different bacterial species, bloodstream infections, foodborne infections, to look at outbreaks and the spread of antimicrobial resistance. And we're also interested in host adaptation, and this is what I'm going to talk about later today, especially animal to human transmission, and how different bacterial clones or bacterial taxa adapt to their hosts. And lastly, I'm really interested in horizontal gene transfer and recombination, and how these processes shape the evolution of different bacterial pathogens. And so if you're interested, here are my websites if you want to know more about our work. And so I'm going to start first with introducing you to our graduate program, to our school. So this is the state of New York, highlighted in red. It's found in the northeast part of the U.S. And if you look closely at in the state of New York, the map of the state of New York, Albany is here. and it's only 2.5 hours travel time to New York City. And other big cities in the state are Syracuse, Rochester, Buffalo, and Niagara Falls. And so there are many, many universities all across the state of New York. And Albany, if you're not familiar with Albany, it is the capital of New York State, not New York City. It is the seat of the governor's office, the Congress, the state Congress. And it is a very interesting history. It was colonized by the Dutch, followed by the British. And now, aside from SUNY Albany, there are many other universities in the area. In Albany, we have the Polytechnic Institute, Albany Medical College, Albany College of Pharmacy and Health Sciences. We have a law school, a Polytechnic Institute, and then 6 private colleges all around Albany. And so here is a really nice picture of the city looking from the Hudson River. If you're not familiar, I hope you will after this. The State University of New York, or SUNY for short, it is the largest university system in the U.S. There are 64 campuses, and these are all marked in this map, all the locations of the campuses. So we have, as I've said, it's the largest university system in the US. There are 14 doctoral institutions. So Albany is one of the 14 that offers a Ph.D. And the whole SUNY system is administered in Albany too. So there's a separate administrative building. Here's the administrative building. So this office manages the entire SUNY system, all 64 campuses. And so, as I've said, SUNY Albany is one of the 14 PhD institutions of SUNY. It was founded in 1844, and we have 3 campuses. Here's the Uptown campus, and our building is it's not shown here, but it's found on the left side of the building of this picture. We also have a downtown campus where many of the social sciences and political sciences programs are located. And then we also have a health sciences campus. So the whole SUNY Albany is divided into these 3 campuses. And so we have more than 50 undergraduate majors, more than 150 graduate programs. And as you can see here, there are many, many fountains, water fountains all across the campus. So I thought I'll include some information about the international enrollment, just so I want to show you that we have a lot of international students. We encourage international students to come. As of 2021, we have representative countries from different regions. These are just a summary of those. All the way from Asia to Africa. And the top 10 countries with the highest enrollment in SUNY Albany is shown here. And so we want to, we'd like to encourage all of you to also consider, so we can increase the enrollment from Brazil. So yes, oops. I think my slide is frozen. Oh, here we go. In addition to different colleges and departments, we also have 35 research centers and 12 research institutes all across the school. And I'm just going to highlight some of them that are relevant to you. We have the RNA Institute. We have centers on neuroscience, X-ray, genomics, global health, public health, what else? Forensics and autism. So these are just some of those centers, research centers and institutes that we have. If you're interested, here's the website for our graduate programs. We have 8 colleges that offers PhD degrees. So we are part of the College of Arts and Sciences. But as if you're interested in any research on microbiology, we have microbiology research not only in the arts and sciences, but also in the public health and also in engineering. So we have, microbiologists working on, for example, wastewater microbes. So here's our department, and our PhD program currently has 41 US students and 17 international students. So we want to increase this number, the number of international students. The program, in terms of the program of study, we have a qualifying exam, in 2 qualifying exams, and we require all PhD students to enroll in 4 core courses plus electives. And we also require 2 semesters of teaching. So usually the first year students are have to teach because we want to improve their communication skills and teaching skills. And then lastly, you have to do a dissertation and public oral defense. We offer both master's and PhD programs. So the PhD program, there are different faculty members working on different diseases. So it's up to you to choose, what you're interested in, from, bacterial diseases, viral diseases, cancer, neurological diseases, and muscle degenerative diseases. In terms of admission requirements, as I've said, I'm a member of the admissions committee. So there's a committee that evaluates all applications. Per year, we usually receive more than 100 applications, but only 6 to 10 are accepted. I know it's a low, admission rate, but we're increasing this over time. So this year, for example, we've increased it to 13 accepted new PhD applicants. If you're interested, the deadlines, and I'm going to talk more about this teaching assistantship, but if you want to be considered for funding, the deadline is January 31st. So you have, 2 months for August admission, and for spring admission, that is, the deadline is October 1st. If you already have your own scholarship, for example, a scholarship from your government or a private scholarship, and you don't want to be considered for funding by SUNY, we have a different set of deadlines, June 15 and December 1. So what do you have to prepare in your application? We need transcripts from all schools. If you already have a master's degree, we also want to see the transcript from your master's, 3 letters of recommendation from your professors, one of which should describe your research experience. And then we want you to, we require maybe 2 to 3 pages of your personal statement where you describe your, what are your motivation, what are your goals, your long-term and short-term career goals. Why do you want to do a PhD, and why do you want to apply in Albany, and which faculty members you're interested in joining. We also want to we require a resume or a CV. And I also want to mention GRE is not required. So you don't have to take your GRE. However, as international students, we require an English language proficiency exam. So it's up to you to choose which English exam you want to take. So first year PhD students, we don't require a master's degree to enter the PhD program. If you only have a bachelor's degree, you're required to take 2 to 3 lab rotations. So you rotate in different laboratories for 6 weeks. If you already have a master's degree, the lab rotations are optional. So you can go directly to your the laboratory you're interested in. Okay, so I want to talk about your teaching and research assistantships. These are what we refer to as funding sources for your from SUNY that supports your entire PhD studies. As I've said, we require 2 semesters of TA for the PhD degree, and then the state of New York will support your teaching assistantship for up to 4 years. So that means if you want to teach for more than 2 semesters, you can do that, and we will support you. We will support you throughout all those years. The research assistantships, or RA, are paid through the advisor's research grants. So you have to talk to your PhD advisor about transitioning from TA to RA. But we also have several PhD students who are paid through the RA. So what do we mean? What do I mean by assistantships, and what do the students get from these assistantships? You get a tuition waiver for throughout your PhD studies. These are the most, I think, last year's number, 2021. But per academic year, the tuition is $25,000, and so that's covered by the assistantships. You don't have to pay the tuition. You can find more information about these tuition fees here in this website. And then we also provide academic year stipends. So we will pay you from mid-August to mid-May. And these numbers are from last year, it is $22,500. So for around 9 months. During the summer, we also provide a summer salary for you. So that's from mid-May to mid-August. And this is $7,500. Again, these numbers have slightly increased this year and will also increase, I think, next year. So all in all, you get all of these benefits from if you're a teaching assistant or research assistant in our PhD program. And then health insurance is subsidized, so we provide health insurance. And lastly, I don't know if you can see it here, the university too also pays for the public transportation. So we have city buses all around the city. You only have to swipe your ID, and you don't have to pay for public transportation. So these are the benefits that you get as a teaching assistant or research assistant in our PhD program. We also offer, because we really encourage our PhD students to learn bioinformatics. So if you're interested, we have this summer workshop. It's a 10-week summer workshop that we've been providing remotely or in person, and it's free. And I didn't write it here, but the benefit for this, aside from learning about bioinformatics, is that if you finish this summer program and you apply to our PhD program, your application fee your graduate application fee will be covered. You don't have to pay. I think it's $75. So you don't have to pay for that as long as you complete this summer workshop. Okay, so I just want to pause before I go to the research part of this seminar and see if you have any questions, or should I wait until the end, Lucia?
Yes, you can wait till the end, Cheryl.
Okay, at the end. Okay. So now I'm going to switch to the research component of this seminar. And I'd like to acknowledge a lot of people, but for today's talk, this research was completed by these individuals highlighted in blue. And also want to mention or thank the funding agencies of our work. So today's research talk, I'm going to describe our work on bacterial population genomics, what it is and why do we study it. And then I'm going to talk about Staphylococcus aureus in humans and animals. Decades ago, more than two decades ago, back in 2002, when whole genome sequencing was in its infancy, this group, Welch et al., sequenced the whole genomes of 3 strains of E. coli, a non-pathogenic, a uropathogenic, and an enterohemorrhagic strain of E. coli. And they just counted and identified all the unique genes that are found in all 3. And they found that throughout the 3 strains, they found a total of more than 7,000 protein coding genes, which is really remarkable. However, when they look at individual strains, they found that there are more than 1,000 genes that are unique to one strain, to the uropathogenic strain. And when they compared 2 strains, they found that there are only 204 genes that are present in these purple strain, the uropathogenic and the enterohemorrhagic, but are absent in the non-pathogenic strain. So this is really curious. And when they compared all 3 strains, they found that out of these 7,000 plus genes, only less than 3,000 genes are present in all 3 strains, which means that there are many, many genes that are unique to 1 or 2 strains. And so this was really remarkable, especially at that time. And so you might think, well, we're only comparing 3 strains. What if we sequence the genomes of more than 3 strains of E. coli? And that's what this other group, Land et al., decided to do. They sequenced the genomes of more than 2,000 E. coli strains. This plot shows on the x-axis the number of genomes, and the y-axis is the number of unique genes. And they found that only around 3,200 genes are shared among 95% or more of the genomes, and that's shown in this red bar. And then just astounding 89,000 genes are unique to 1 or few strains, shown here in blue. So again, this is really mind-boggling. And if you put all these genes together, this is what we refer to as the pan-genome, the totality of all the genes that are found in a population or in a data set of genomes. And so, what this means is that, and this has not been, what this means is that not one genome or strain can truly represent a species. And similar patterns have been reported in other species of bacteria, not just in E. coli, whether it's their environmental bacteria or clinical bacteria. And this is where the field of population genomics come in. The study of hundreds and comparison of hundreds and thousands of genomes of a single species. And so we ask the question, what makes them different? All these strains of the same species, we look at mutations and SNPs and recombination and indels and mobile genetic elements and accessory genes. And equally important, we ask the question, to what extent are these differences clinically relevant? So we use all this genetic variation between strains to answer questions or to study virulence and adaptation, antimicrobial resistance, to map disease transmission and outbreaks. So this really sums up our work in bacterial population genomics. So there are many methods used in population genomics, but what I'm going to mention today is the genome-wide association study, or GWAS, which is now becoming more and more frequently used in population genomics. But this method has been it's an unbiased screening approach that has been widely used in human genetics to study genetic variants that are associated with a phenotype. And usually in human genetics, you divide your patients or your individuals into your disease cases versus your healthy individuals, and you sequence their genomes or certain genes, and you look at SNPs, usually SNPs in human genetics, shown here in red, and then you calculate the frequency of those variants and how they differ between your cases and controls. So as I've said, this has been widely used in human genetics, but we're only now using this method in bacteria. And in bacteria, it's quite different because we're talking about clonal organisms that divide clonally, and so we have a slightly different method of you in bacterial GWAS. So you start with a mix, multiple strains. You divide them into two phenotypes. So in this case, let's say one group of strains are exhibit one phenotype, let's say drug susceptibility, versus another group of strains that exhibit drug resistance, for example. And you sequence all the genomes of all your strains. Again, we're only looking at two phenotypes, and then you visualize the output of your GWAS, and I'm going to talk more about this in a later slide. So you calculate the frequency of your variants, and then you do some validation tests using statistics and experimental work and phenotype testing and so on. As I've said, you start with usually hundreds of genomes. You sequence their, you look at their sequences and look at the, genetic variation between them. So let's say you look at both homologous recombination like the SNPs or non-homologous variations such as indels, and you calculate the probability that these variants are associated with your phenotype, in this case drug resistance versus drug susceptibility. So Staph aureus, and I'm sure many of you know already, is normally carried on mucosal surfaces and skin. It's usually harmless, but they can cause serious infections, such as some of these infections, severe infections, are shown here. So it is a very important bacterial pathogen. But interesting about Staph aureus is that it can infect many hosts, not just humans. This figure shows is a summary from a paper where they look at transmission of Staph aureus between different hosts, animals and humans. The arrows indicate the direction of transmission, and the width of the arrows indicate the frequency of those transmission. And previous studies have shown that some Staph aureus clones are specialists. That means they're likely to infect only one host species. However, other clones are considered generalists, or they are able to infect multiple host species. And I'd like you to remember this, because I'm going back to this statement, because I'm going back to this at the end of my talk. So some clones are specialists, some are generalists. As you can see in this figure, humans are major exchange hubs or transmission hubs. And mainly because humans are always interacting with many of these domestic animals. So we want to ask the question, what bacterial factors contribute to the transmissibility and infection of Staph aureus in human and animal hosts? So we used two data sets. One came from human patients with bacteremia. This is in collaboration with the Dartmouth Hitchcock Medical Center. We collected one isolate per patient, and we have a mix of methicillin-resistant and methicillin-susceptible strains that we have collected from 2010 all the way to 2018. And then our second data set is from diseased animals. These were collected by the New Hampshire Veterinary Diagnostic Lab from 2017 to 2019. Most of the Staph aureus, again, these are all from animals diagnosed with disease. Most hosts are cats, dogs, and cows, understandably. But we also have a few from, lizards and rabbits and rodents and goats and so on. Again, we have a mix of methicillin-resistant and methicillin-susceptible isolates. So we combined all of these two data sets from humans and from animals, and we have 437 genomes in total. Again, this is a summary of those isolates. Three quarters of them came from humans, and then everything else from animals. These came from different states in the northeast part of the US, indicated by this blue. Again, we have a mix of methicillin-resistant and susceptible isolates. But I want to mention that we have an imperfect data set because we have an uneven numbers of animal and human isolates. We have more human isolates. We also group all animals together, even though we all know that they're very different from each other. And the sampling period partly overlaps, so we cannot be 100% certain that there was direct transmission between humans and animals. But regardless, we were able to, as I will show you earlier, we were still able to, do a robust analysis on this data. So the first thing we did was to identify our clones, Staph aureus clones that are present in our data set. We identified 7, and these are indicated by CC and highlighted in colored boxes in this phylogenetic tree. So we have 7 dominant clones throughout the population. And when we look at individual clones and how they're distributed in different hosts, we found, so this figure shows the 7 clones in the x-axis and the proportion in the y-axis. For each clone, we look at the proportion of isolates found in animals in black and in humans in yellow. And you can see here that all the dominant clones are found in both humans and animals. However, we found significant differences. These two clones, 45 and 8, are significantly associated or frequently found in humans. And we have another clone that is more frequently found in animals. But regardless, all the dominant clones, as I've said, are found in both animals and humans. For each clone, for each of the 7 clones, we look to see if there's evidence of transmission between animals and humans. So, for example, this is clone 1, CC1, and each circle represents a genome or isolate. And if it's blue, that means it came from human, and green, light green, means that it came from blue for animals and green for humans. And so we were able to map the transmission within each clone between animals and humans, and we identified a total of 15 transmission clusters, and these are shown here. And so for example, we have all these animal isolates that are infecting humans too. And so you might think, okay, these networks are really, they look great. But we also did some calculations to quantify these transmission. And I'm not going to go on the details of these calculations, but we calculated that host switching or host infection occurred more frequently from animals to humans, so from blue to green, than from humans to animals. So again, more transmission happening from animals to humans than from humans to animals. Okay, let's now go to GWAS. I mentioned earlier that I'm going to briefly talk about how to visualize your GWAS results. And this is referred to as Manhattan plot. In a Manhattan plot, you plot the p value of each genetic variant on the x-axis, on the y-axis, against its position in the genome on the x-axis. So each dot in this plot represents a genetic variant. It can be a SNP, it can be an entire gene, or it can be just a DNA segment. And each gene or each genetic variant has its own p-value. And since there are, thousands and thousands of these genetic variants and therefore p-values, we would average, we would use a genome-wide significance of those p-values, and that's indicated by this blue horizontal line. So that's the genome-wide significance value on which we consider all these genetic variants. And so that p-value would indicate the difference in the frequency of the variant in the two phenotypes you're testing. So anything above that horizontal line would be considered statistically significant. So we want to look at all those genetic variants above that horizontal line. So again, to reiterate, we have 437 whole genome sequences, 114 from animals, 323 from humans. We carried out 3 different GWAS analyses, one based on SNPs, one based on K-mers or short nucleotide segments, and one on the presence absence of accessory genes. And I'm not going to show you all the details of all 3 GWAS, but just to give you an example of what you found. So this is a GWAS, a Manhattan plot for one of the GWAS analyses we did, and we found that for all 3 GWAS, based on SNPs, K-mers, and accessory genes, we found significant association with a prophage, a Phe-SA3 prophage. Again, we want to look at those dots that are above that horizontal line. And if you look close, if you zoom in on that location, you're going to see these different genes on that location, and many of them are hypothetical, but those with known functions, we found that they are related to human immune evasion genes. So these are the 4 genes with known functions. We have a complement inhibitor, which inhibits opsonization and phagocytosis. We have a staphylokinase, which converts plasminogen into plasmid. We have a hydrolytic enzyme and ATPase. However, you might think, if you remember, I said our data set is, we have more human isolates than animal isolates. And so you might think maybe these results are affected by the uneven numbers of animal and human isolates. And so we did a 4th GWAS method. If you look back at our big phylogenetic tree that has all the 7 clones, what we did was we took a subset of that big data set, whereby we only look at branches where there's always a pair of closely related animal isolate in red and a human isolate in yellow. So we have a smaller phylogenetic tree that has, again, a pair of animal and human isolates. So if you look at each branch, you'll always see a pair of them. So here now we have an equal number of animal isolates and human isolates. So we did a GWAS on this. So we have a total of 36 pairs. And I'm not going to show you all the details, but we did find the same genes that we found earlier, as well as 17 additional loci that are associated with infection of human versus animals. Okay. So again, we carried out 4 different GWAS methods, and we identified these genes, especially these 3 that are associated with human and animal infection. So this is more evolutionary biology, but we found that genomic variation associated with infecting animals and humans is under strong natural selection. So what we did was, if you remember your genetics, we have a narrow sense heritability. This is the only type of genetic variation that natural selection can act on. And we calculated the heritability of the genes we found, the SNPs we found, and the K-mers we found in our GWAS. And we found that, so for example, in the SNPs, this orange bar, we found that 88% of the heritability is associated with human and animal infection. So that means the ability to infect humans versus animals can be explained by 88% of this genetic variation. And out of this 88% heritability, 99% of that is attributable to the prophage genes that we identified. So this means that if you think about it, if a pathogen, is transmitted from animals to humans, that change in the host can impose a very strong selective pressure on the bacterium. And this genetic variation that we're finding is driven by this prophage genes that are causing this infection of animals and humans. And then lastly, our last analysis is that this prophage locus that has all the immune evasion genes, we found that it's a hotspot for genetic recombination. And I'm just going to show you a small snapshot of that. We analyzed the recombination of the entire length of the chromosomes, as well as all the strains, but I'm only going to show you a subset of that. This is one of the clones. Here's the phylogenetic tree of that clone, and this is we show here the host type, whether it's animals or humans, and this matrix shows the regions in the genome that have been recombined in red in colored boxes. And you can see that, and the gray boxes indicate that these are genomic regions that have not experienced any recombination. And you can see here that within that prophage locus and the neighboring regions, there's very high frequency of recombination that we've identified. And so just to summarize, what I've shown you today is that there's a lot of transmission happening of staph aureus between humans and animals based on the network analysis and calculations we did. Based on the GWAS, we found that host transmissibility and infection are driven by the genes in that prophage. And then lastly, that prophage is spreading across the population through recombination between strains. And if you remember earlier, I asked you to remember what I said about previous studies saying some Staph aureus clones are considered specialists and some are generalists. But what we propose in this, or what we were seeing in this study, is that any clone of Staph aureus can rapidly evolve and become adaptive in both animals and humans upon acquisition of that prophage, including those typically considered specialist clones. In other words, we think that there's no such thing as a very strict as a strict specialist clones. Any clone can be will has the ability to infect both animals and humans based on the presence or absence of this prophage. And so if you're interested to read more about this work or learn more about this work, especially the methods, this is already published, and it's open access. So you can just download the PDF. And that's the end of my talk. So if you have any questions, I'm happy to answer them.
Well, Cheryl, the audience can only make questions through the chat.
Okay.
Okay.
Yeah.
And they can be done either in Portuguese or English, and then we'll be glad to translate it.
Okay. So there's a lot that says bom dia. What does that mean?
Good morning or good
Good morning.
afternoon. Good afternoon in the day.
Okay.
That's
So those people who are watching, if you have questions about the graduate program or the teaching assistantships or the research, I'm happy to answer them. You can also email me later on if you want, a more private, discussion.
yeah, that's a good point, because the video will be available so people can see it anytime and check on the instructions that you gave already and the points to make contact. It's important to check that you have shown um, that probably the best way to apply for either PhD degree or a postdoc position, the best way is to contact first the investigator, right?
Yes.
Okay.
So
Okay.
yeah.
People don't need to go to the academic administration first,
No.
right?
So I actually receive around between 30 to 50 emails asking me if I'm from students all over the world every year, up to 50 inquiries about whether I'm accepting PhD students, and I try to respond to all of that. And I want to tell everyone that um, what we look for as part of the admissions committee, what we look for is whether there is a fit, there's a good fit between the applicant and the faculty, whether we can find an advisor for the applicant.
Okay.
So
So
even
first thing is looking for an
yes,
advisor, the
yes,
right advisor.
yes. So go to the faculty website. There are the list of faculty members and their research interests or research background. See if there's anyone there that you're interested in, and then send them an email. I cannot guarantee that they will all respond to you, but send an email asking whether they're accepting new PhD students in the next academic year or not, because sometimes they don't. Maybe they're already full. Maybe uh, you know maybe they're going to retire. So you have to ask first. um, And then send your CV and tell them why you're interested in their research and so on. What I usually do is if there's a student who emails me and I really like the CV, I will have a very informal Zoom conversation. We just talk about, to see if we have a good fit. And then apply if you think you can, you're really interested in that lab. There is a question from Deborah Neri. Is there an age limit? No. So you can apply while you're still in your undergrad, you're a senior. And so again, we don't require a master's degree. That's quite common in the US. You can go directly from your bachelor's to your PhD. But what we're looking for is research experience, not just grades. So if you volunteered in a lab, or you've probably worked after graduation for a few years, we want to see that you actually are interested in research. So, and we have different age, we accept age is not a requirement.
Okay. Well, Victor Leite, who is a PhD student in our lab, he's congratulating you
Oh, thank
for the
you.
very nice work. And the results are quite interesting. I'm sure he was looking for the drivers in staff areas. In fact, I have a question for you
Yeah.
about the staff areas issues. Um, you have presented the classification in CCs, in clonal complex. Is that based on the MLST derived from whole genome analysis only, or have you added the additional set of genes?
The MLST, just based on the
The
MLST
basic MLST for SRs, right?
for the CC, but the whole phylogenetic tree is based on the core genome.
Oh, okay.
Yeah.
Okay.
So
Very
Deborah has another question. There are positions available for postdoc. In fact, just a few months ago, I just tweeted, that I'm looking for 2 postdocs, and unfortunately, I'm sorry to say that I've already accepted 3 new postdocs. So maybe, uh, or we can talk. Send me an
yeah.
email, Deborah.
But according to something that you have said, somebody can apply for a postdoc position if having all the support from their own country, right?
Yes. So if you have your own fellowship, like a
Yes.
Fulbright, yeah,
Okay.
yeah, that's
Or
absolutely.
for the country or any other
Yes,
institution. Okay.
yes.
Okay.
So,
Good. Good.
mhm.
So, any additional questions from the audience?
I just want to mention too that all the PhD applicants that we admit, that we
Mm.
accept, are guaranteed funding support. If you don't have any, and
Oh, okay.
yes,
Okay.
so we don't
Okay.
accept anyone that we are
There's
unable.
no support at all.
Yes, uh,
Okay.
yes.
And how about the benefits from SUNY?
What kind of benefits? What?
Um, like health insurance
Yeah.
or things like that?
Yes. So I mentioned that earlier. Health insurance is covered. You should get health, dental, and vision
Uh-huh.
insurance. And also mentioned that public transportation is also covered, so you don't have to pay.
Okay, yeah, that's something very good.
Mhm. Yes,
So the students can go around without
yes.
paying any
It,
fee.
it
Right.
can be very expensive.
Yeah.
Yeah.
That's probably okay in at Albany, but not in New York City, right?
Not in New York City. It's more expensive there.
So you better be at Albany.
Yes.
Okay.
And yeah.
So let's see if we have some other question. I don't think so. So it's good to reinforce that the presentation will be available
Yeah.
at any time from YouTube. And I would like to congratulate you for the excellent presentation, quite informative, quite enthusiastic. I'm sure many students are now um,
Yeah.
would be delighted in being in your lab or in other
Mhm.
health laboratory
Uh,
at Albany. And let's see if you have applications for the near
yes.
future.
And again, I want to reiterate that the summer bioinformatics workshop, the 10 weeks, that's free. If you
Mhm.
complete that and you apply for our PhD program, the application
Uh-huh.
is waived.
Yeah,
Uh,
that's an additional opportunity
yes.
which I found it's very interesting. So
Yes.
thank you so
Thank
much,
you.
Cheryl, and we certainly be in touch.
Yes. Thank you very much.
Okay, see you soon on the screen, I guess.
Yeah.
Thank you so much again.
Bye.
Bye bye.